Hello everyone and welcome to today's webinar, High-Throughput CRISPR Editing Using the Onyx Platform Identities Essential Residues in Proteins. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. Now we encourage you to participate today by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speakers, Dr. Liselot Dewachter, Postdoctoral Research Fellow, and Dr. Laura Klitten, Scientific Liaison at EMEA for Inscripta. Welcome to both of you. Dr. Klitten, you may now begin your presentation. Welcome.
Thank you very much to LabRoots for giving me the opportunity to introduce to you the Onyx platform and how Inscripta enables genome engineering on a massive scale. I'm Laura Klitten, the Scientific Liaison at Inscripta in EMEA, and I act as the link between scientists in Europe and my colleagues in the US. Inscripta's goal is to empower scientists whose gene editing research is stifled by current technical and licensing limitations, and by providing our unique platform and engaging in collaborative business practice, such as making our MAT7 CRISPR nuclease free for research and development purposes, we enable scientists to realize a new era of biological discovery. We are headquartered in Boulder, Colorado, with offices in Pleasanton, California, and San Diego in the US. Inscripta is backed by leading investors including Venrock, Foresight, Paladin Capital Group, and Melior Development. Here in Europe, I'm your local contact person, so please feel free to reach out to me by mail, phone, or social media if you have any questions whatsoever. I've listed my Twitter, LinkedIn, and email so you know where to find me. So at Inscripta, we strongly believe in the power of biology to change the world. And what we've seen over the past decade is that biology has become increasingly important in all of these areas, from engineered strains that produce antibodies or small molecules to engineered strains that produce the key ingredient of the impossible burger, to other strains that break down plastics or other byproducts. And this trend is just going to continue, we believe, over the next decade. So really, our ability to understand how biology works and use that knowledge to engineer biology to create new desirable phenotypes, we believe is going to drive advancements in all of these sectors of the economy. And a key thing to both understanding biology and engineering biology is our ability to manipulate genomes, and in particular, our ability to manipulate genomes in high throughput. So there are a number of current approaches for doing high throughput genome manipulation. So you can do classical mutagenesis, knockout libraries, or CRISPRi or CRISPRa libraries. But all of these approaches, while very beneficial in certain scenarios, they do come with some trade-offs. And those trade-offs are depicted over in this plot on the left. So either an approach will typically allow you to assess a lot of locations in the genome, for example, when you're doing classical mutagenesis or genome-wide knockouts. Or it will allow you to make a diverse range of edit types if you are doing, for example, protein or pathway engineering. But it's difficult to target both a lot of genomic locations with a lot of diverse edit types in the same experiment. And there are additional challenges with these current approaches. So they suffer from lack of precision, they can have slow or laborious workflows that often require substantial automation to achieve high throughput. It can be difficult to separate hitchhiker mutations, and if you're working on a plasmid, then you can have issues with transient expression and changes due to the marker or the expression system itself. So what we really want to unlock is this upper right quadrant here and also overcome these different challenges to have a reliable high throughput method for manipulating genomes to advance all of the before mentioned sectors of our economy in the future. And that is where Inscripta comes in with our Onyx platform. So this platform enables diverse edit types, genome-wide, and it has a barcode-based tracking system for easy experimentation. So this is the Onyx digital genome engineering platform. It's not just an instrument. It's a whole platform that comprises of the instrument, the consumables, the assays to measure phenotypes in your experiments, and also software to design the edits upstream and to help you interpret the results downstream. And this is a look of how the Onyx platform enables researchers to rapidly cycle through these experimental cycles that we call DGTL, or design, generate, test, and learn. So starting with design on the upper left corner, the Inscripta designer software enables you to design thousands of edits that you want to make in your experiment based on algorithms that are optimized the way these edits are being delivered into the genome. After you create an order in the software, it kicks off a manufacturing step at Inscripta, where we generate a custom set of ready-to-use consumables that are shipped to you, together with our proprietary microfluidics cartridges, and you load these onto the deck of the Onyx instrument together with your cells. The instrument will then run your entire wet lab part of the experiment for you. The input to the instrument are these microfluidics cartridges, your custom designed library of edits, all the media and buffer needed, and then also cells. You can use Inscripta standard cells that we provide with our kit, or you can use your own custom strain as an input to this editing process on the instrument. The output from the Onyx instrument is a population of engineered cells that we call the engineered cell library. And let's just zoom in and look at what this is. This is a diverse pool of edited cells. There is a single cell per edit, and the whole library, there can be up to 6,000 edits for yeast and up to 10,000 edits for E. coli. And the edits can be diverse. They can be insertions, deletions, or substitutions, up to 63 base pairs in length. And there are also barcodes on the plasmids that enable you to easily track enrichment and depletion in experiments downstream from this pool of edited cells. So what you do is you take these cells and you can use them in a diverse range of experiments that you want to run. You can either plate them out and pick single colonies and put them into, for example, 96-well plates to do a direct measurement of a metabolite or small molecule, for example. You can use mass spec or fluorescent-based readout, or you can also keep the cells in a pooled format and run a selection where you inoculate a flask with the pooled library that you subject to some conditions under competitive growth, and then you can track enrichment and depletion of individual designs using the barcode system. And then after you run your experiments, we have assay kits that help you prepare NGS samples for sequencing. And after the sequencing, the raw FASTQ files can be analyzed in our Inscripta Resolver software, which allows you to link genotype to phenotype and learn from your experiment, which can then be feed into the next cycle of GTTL, so the next cycle of experimentation, enabling you to design new designs based on what you learned from your first experiment. So here are some key features of the Onyx platform. It works with both Saccharomyces cerevisiae and E. coli. You can use wild type or custom strains. The key innovation of the technology is that it uses cell-based partition and microfluidics to enable these large-scale libraries, and with really high coverage of all of the up to 10,000 designs that you have in your library. It has an optimized editing chemistry, including our proprietary MAT7 nuclease, and the software relies on really advanced design algorithms to enable delivery of diverse edit types to the genome with high efficiencies. And the Resolver and Designer software are really easy to use, but they're powerful. And the instrument itself is push-button operation, so it's really easy to do these cycles of GTTL with just one or two researchers running the entire cycle. So let's dig a little bit more into some of the key features of how the Onyx platform works. So one aspect of the platform is the Onyx chemistry. Normally, when doing CRISPR experiments, researchers would have to separately add the guide RNA to guide the nuclease to the correct locus on the genome, and then also add a donor molecule to drive template-mediated repair. So in order to make sure the right guide RNA and donor molecule are being transformed into the same cell, you need to partition these into, for example, 96-well plates. But that really limits your throughput if you are limited by the plate format. So for the Onyx platform, instead we use the cells themselves as the partition. So this enables you to have thousands of partitions in a single experiment, and the way that this is accomplished is by covalently linking together the guide RNA guiding the nuclease, the donor template sequence, and a barcode to enable easy tracking in your experiments. I just want to highlight here that it's only the donor sequence which is incorporated into the genome. The rest stays on the plasmid. So now for generating these large and diverse libraries of different edit types, each with different efficiency on a single cell format, we had to develop a lot of novel technology, in particular microfluidics, to keep the uniformity of distribution of the designs in your cell population during the editing process. And there are also some other components to the Onyx chemistry, including the guide RNA, the donor, and the barcode. Obviously, we have the MAT7 nuclease to actually make the cut, and then repair proteins to help the cell's repair mechanism. They're also supplemented as part of the Onyx chemistry. Another key feature of the Onyx platform is the easy-to-use software. And I'll just show you one example of that, and that is the Inscripta designer software, which is shown here. And there are two ways that you can make a set of designs for a library that you want to use in your experiment. One is by using the graphical user interface, which is easy to learn, but it's very powerful. It allows you to make substitutions, insertions, deletions, or do a full saturation mutagenesis on either a single gene, a set of genes, or all genes in the genome with just a few clicks. You can target specific locations like inside genes, in front of genes, or behind genes. The second way to create a library is by using the CSV file upload. This enables you to make all the same types of designs, and it's friendly with bioinformatic pipelines. So if you want to use scripting or informatically design your libraries, that is also very easy to do. So the software is really easy to use, but it's very powerful because it's driven by robust algorithms that power high editing efficiency for all of the edit designs that you want to make on the genome. And this is driven by machine learning models that are built on very large empirical data sets that we have collected at Inscripta. So these models have been trained on more than 300,000 designs with thousands of features evaluated in the model, and we are continuously improving these models over time. So in total at Inscripta, we've created more than 3 million designs and optimized all of the aspects of a design, the guide, the donor, the barcode that go into the cell to deliver the edits that you want to make with a really high efficiency. And this is one of the keys of the Onyx platform, is that you can make these diverse edit types. So you can do any edit that is between 1 and 63 base pairs in length, insertions, deletions, or substitutions. And with these algorithms that power our design software, we are able to target more than 95% of the wild-type genome. So that means that you can target coding regions with things like a triple-stop substitution to knock out genes, or do a full saturation mutagenesis of amino acids. And you can also target non-coding regions to make regulatory changes in the genome, like inserting a synthetic promoter, RBS, in E. coli, or synthetic terminators in yeast. So these are just some examples. And there are essentially infinite ways that you can manipulate the genome with this technology. I've just listed a bunch of ideas here, but what I really want you to think about is you could do genome-wide truncation, saturation mutagenesis, like we already talked about, and we'll also see an example a bit later. But there are also many ways to do transcriptional or post-transcriptional regulation. So what I want you to start thinking about is, what types of libraries would you design if you had access to a technology that would allow you to manipulate the genome with this scale and ultimately help you understand how biology works better, or engineer strains with the phenotypes that you need. So here I show some real-life examples of projects we've run internally or together with a few of our early access partners. We've made it easy to alter gene expression with insertion of a promoter ladder in front of every gene in the E. coli genome, as well as functional knockouts by inserting triple-stop codons to generate truncation of proteins. We worked on transcription factor binding site substitutions in yeast in order to alter transcription and thus expression of the genes. The Onyx platform makes saturation mutagenesis of single proteins or even complete pathways very easy, as we can do up to 10,000 edits per instrument run. And in a minute, you will actually see a real-life example of how that worked. We've also shown with Jeff Bouquet from NYU how our technology can be used to quickly debug strains in working on creating these synthetic genomes. And finally, we've shown that Onyx technology works on improving heterologous gene expression and understanding in yeast. So I just want to end my talk with asking you to think about which experiments you would perform if you had access to this kind of technology. And I thank you for attention. And now I'll turn it over to Liselot Dewachter, who will present some amazing data created on the Onyx platform.
Hi, indeed, I have been able to test this technology at a very early stage as an outsider, and will share with you my experiences. So if you're listening to this talk, it means that probably you are a life scientist. And if you are doing research or you have done some research in the past, chances are that your project would have looked something like this. You wanted to investigate your favorite biological phenomenon, and you started with some sort of screening strategy. Now, because obviously your screen was well thought out and it was executed to perfection, you ended up with a nice number of significant hits. So the next step was then to prioritize these hits, think about them deeply, and then pick your favorite one. Now, this favorite hit you chose is probably a protein that is not well characterized, and it is now your life's mission to fully characterize everything. So you know this protein is involved in the process you screened for, but how? Does this protein perform a catalytic function? And if so, where is the catalytic site and which residues are involved? Perhaps there's an important protein-protein interaction, and you need to know which residues mediate this interaction. You even might want to optimize or change the protein in some way, but also for this, you would need to know which residues are involved in its function. And what you may want to do to figure this out and start to answer these research questions is to randomly mutagenize the associated gene, for example, by error-prone PCR. However, there are some important limitations to this strategy, because error-prone PCR by definition is random, meaning that you have no way to control which mutations you will introduce. And there's therefore no way to be sure that you have targeted all residues in your protein, and some information may be missed. Secondly, by doing error-prone PCR, the mutational space that is available to you is quite limited. Because with this approach, you will introduce usually only one mutation per codon, meaning that the amount of amino acid substitutions that are possible is limited. Take this example shown here on the slide with a lysine residue. Introducing only one point mutation makes it possible to change to 6 different amino acids, meaning that the other 13 are unavailable to test. So also here, a lot of information is lost. In an ideal world, you would systematically target your entire protein to be sure that you have tested all residues. On top of that, you would replace every residue with all different amino acids, because some amino acid substitutions behave completely differently. So in short, if you would perform a full saturation mutagenesis on your protein, only then will you be sure that you have all information. But I think I do not need to tell you that these are a lot of mutations to introduce. If you want to saturate just one position, you already need 19 mutations. And if you want to do this for an entire protein, the number of mutations you need is 19 times the length of your protein, meaning that even for short proteins, the number of mutations easily rises above 2,000. Now, until recently, this really was not worthwhile. No one in their right mind would, I think, just set out to create 2,000 different mutations. But fortunately, today, this is within the realm of possibilities, thanks to efforts like the Onyx platform that use high throughput CRISPR editing to generate thousands of mutations in parallel. And just to make sure that we are on the same page, how the Onyx platform does this, at least in E. coli, is by introducing two different plasmids. One plasmid encodes the MAT7 nuclease that will cut the DNA, and the other plasmid introduces a very short guide RNA and a rescue oligo. Now, the rescue oligo is homologous to the genome and will integrate into the genome based on homologous recombination, and it also contains a certain mutation you want to get in there. Now, it's of course easy for any company to claim that they have developed this brand new technology that is capable of generating thousands of mutants. We were wondering if it is actually true. So we wanted to test this technology and see if we could use it to perform saturation mutagenesis, and if it was successful, what kind of information we could get out of these saturation mutagenesis libraries. So we chose 4 different proteins in the bacterium E. coli to mutate. These are LPXA, LPXC, MUR-A, and FABZ. And all of these proteins are involved in the synthesis of the E. coli cell envelope shown here on the left. The inner layer of the cell envelope is the inner membrane, which consists of phospholipids. And phospholipids are created, of course, in a multi-step enzymatic process, of which one of the steps is catalyzed by FABZ. The inner membrane is surrounded by the peptidoglycan, which is a rather thin but very rigid layer. And peptidoglycan is produced by, among others, MUR-A. Then the most external layer of the cell envelope is called the outer membrane. And the outer membrane also contains phospholipids, but besides that, it also has an outer membrane-specific component, lipopolysaccharides. And lipopolysaccharide, or LPS, consists of a core structure which is called lipid A, and this structure is created by LPXC. We then also added the LPXA protein, which catalyzes a reversible step that produces and consumes all of the substrates for these 3 different enzymes. And LPXA is therefore thought to act as some kind of hub in the synthesis of the different cell envelope layers. Now, why did we choose these targets? Well, there are several reasons. First, they represent a nice mix between more and less well-characterized proteins, meaning that there is information available in literature that we can use to validate our findings using this novel approach, but also that there is still some new discoveries to be made. And a second reason, and perhaps more importantly, is that all of these proteins are involved in cell envelope synthesis. And historically, the cell envelope has proven to be one of the most effective antibiotic targets out there. And you may or may not know, but we are currently on the verge of a worldwide antibiotic crisis, because more and more bacteria are becoming resistant to almost all or even all our current antibiotics. So we urgently need new compounds to fight even the most resistant bacteria. And in this context, a lot of effort is being made into discovering inhibitors of mostly LPXA and LPXC. These are considered very attractive targets. So we want to fully characterize these proteins and try to help this effort. Now, in our saturation mutagenesis libraries, we did not just replace every amino acid with every other amino acid, but for all of these positions, we also included a synonymous mutation. So this synonymous mutation should not have an influence at the protein level, but it does introduce a change in the DNA, which we can track through sequencing. And so for us, this is an important quality control, which I will discuss in a few minutes. But this means that for every residue in the proteins, we have created 20 different edits. And this brings us to a total of almost 23,000 different edits that we were able to create just like that. And how did this practically work? Well, we created the 4 different libraries completely separately from one another. For all of them, we started from the same E. coli wild-type bacterium, and the correct plasmids were then transformed, of course, with this microfluidics platform that you have heard about. And this generated for us the different mutated libraries. Now, of course, because CRISPR editing is not 100% efficient, our mutated library still contains some wild-type cells, but fortunately, we also have a high enough percentage of edited cells that contain the desired mutations. What we then did with these libraries was simply sequence the genes that were supposed to be mutated to detect which mutations were actually present. And at this point, it's very important for you to know that all 4 of these proteins and their associated genes are essential for E. coli viability. So this means that if we introduce a mutation, for example, in LPXA, that abolishes protein function, this specific mutant will die, and it will disappear from the library, and we will not find it when we sequence this gene. On the other hand, if we introduce a mutation that does not interfere with protein function, this mutant survives, we sequence it, and we detect the mutation in question. And so by looking at which edits are and are not present among our sequenced reads, we hope to be able to define which residues of these proteins are essential for their function. But before I can address this research question, we were, of course, wondering if the saturated mutagenesis libraries that were created by the Onyx platform were of sufficient quality to try and answer this question. So as a first step, we did some quality control. And the first thing we did was look at if there were any residues in these proteins that simply could not be targeted by the technology. For example, because PAM sites were too inefficient or any other reason. And these uneditable residues, so residues that cannot be changed to any other amino acid, but for which we also could not introduce a synonymous mutation, are indicated here with white lines. So unfortunately, we do find uneditable residues that simply cannot be targeted, but fortunately, their number is quite low. We have a maximum of 5 different uneditable residues that cannot be mutated due to technological reasons. And I say it's a maximum because I actually believe that some of these residues are in this list simply due to biological reasons. For example, if you look at the methionine at position 1 in the MUR-A enzyme, I think this is purely biological that it occurs here. Because of course, this methionine is encoded by a start codon, and in this case, the start codon that is being used is the most efficient one, meaning that any mutation of this codon would lead to either a complete inhibition of translation or at least a lower translation efficiency. And so a lower amount of the MUR-A protein and therefore a lowered viability. But so I have, of course, no way to prove that this is the case. So to be on the safe side, we say that these residues are uneditable. Still, I think 5 is a reasonable number to work with. So we then went on to the next stage, which was to ask ourselves the questions if these libraries are actually fully saturated. Now, fully saturated would mean that all of the 19 amino acid substitutions that we created per position would also be created and would be introduced into the genome, meaning that the maximum level of 19 would be reached here throughout the protein. But of course, for an essential protein such as LPXC, this is not what we expect. Because even if we are able to introduce some of these mutations in the genome, they will disappear because they do not support viability. So in the case of essential genes and essential proteins, this is not the way to evaluate saturation. So to do this in a different way, we looked at only synonymous mutations. The reason being that synonymous mutations should not change anything at the protein level. So in theory, we should be able to introduce them into the genome without affecting viability. Meaning that if a synonymous mutation is absent, it's probably because the library is not fully saturated. And so here you can see that the synonymous mutations that are absent, represented by white lines, do quite frequently occur. If we compare the number of missing synonymous mutations with the number of synonymous mutations that we do detect, we'd see that for all libraries, the saturation level is around 90%. So definitely it's not perfect. Perfect would be 100%, but still 90%, I would argue, is quite high. What does it actually mean, this 90% saturation level? Well, it means that the probability that any one specific mutation is missing from the sequencing pool just due to random chance is 10%, and this is quite high. But remember that for every position, we designed 19 different amino acid substitutions. And the probability of all amino acid changes missing for one specific position actually becomes very, very low. It's 10 to the minus 19, meaning that this will not just happen by random chance, and that if we find positions for which this is the case, there's most likely a biological explanation. So I believe with this 90% saturation level and the numbers I have presented just now, I think it's safe to say that we can use these libraries to try and determine which residues are essential for protein function. And this is exactly what we are going to do now. So to try and not bore you to death, I will limit this discussion to only one of the libraries, the LPXC library. And just as a reminder, LPXC catalyzes the first committed step in the production of lipid A, which is an essential component of the outer membrane of E. coli. And what you see here in this big plot is basically all information we have for this library. On the x-axis, you see the position in the LPXC protein, and on the y-axis, you see by which amino acids this position can be occupied. If there is a white line, it means that this specific amino acid could not be found at this specific position. If the line is blue, it means that we did find it. And the darker the shade of blue, the more frequently we encounter this. And now what we are looking for are positions in LPXC for which white line occurs vertically, because these positions cannot be changed to a lot of amino acids. Now, if you squint your eyes a little bit, I think you will be able to see a very clear one in between position 180 and 200. And if you would look in more detail, there are a couple of others that you can discover. But to make your life and also my life more easy, let's look at the data like this. So what you now see on the y-axis is the number of successful amino acid substitutions. If this number is 19, it means that at this position, all different amino acids are tolerated. And this plot, it's more easy to see which residues are actually essential. But this brings me to, I think, rather a complex discussion, because if we want to define which residues are essential, we need a definition for essentiality. And I think this is something you can discuss about, and I think several different definitions can be put forward. But something I believe no one could disagree with is that a residue must be essential if it cannot be mutated to anything else. And I believe this is the most strict definition of essentiality that you could come up with. And the residues that conform to this definition, we call them absolutely essential. And these residues are the ones that have a value of 0 in the plot on the left. So here in this table, you can see all of these absolutely essential residues we define with our new approach. Of course, the first question is, are these residues indeed essential? And so to answer this question, I dived into literature and looked what was already known for the LPXC protein. And here, shaded in gray, are the residues that we know from literature are indeed essential. So hooray for us, because indeed a lot of the residues we identify here are, of course, correct. And at this point, I want to discuss the last residue in the list a little bit further, this H265 residue. Because if you have been paying incredibly close attention, you might remember that this was also one of the uneditable residues I discussed before. And so also for this residue, I now believe that there is a biological explanation why we find it as uneditable. We know from literature that it is essential, explaining why no amino acid substitution could be found. And I believe the reason that we do not find this synonymous mutation for this 265 residue is simply because of pure luck. Remember that the chance of any one specific mutation missing from our library is as high as 10%. In any case, I think this list shows that indeed our approach is capable to capture information that is biologically relevant. And importantly, we also identify some new residues here, residues that were not previously known to be essential. And this is very exciting to us. Of course, this leaves the question, are these new residues indeed essential? And so to try and build some confidence in our method, we plotted these residues on the LPXC crystal structure. And then you see that all of these absolutely essential residues shown on the protein in blue, they all group together in and around the protein's active site. So this gives us a lot of confidence that also the newly identified residues are indeed essential for protein function. So far so good, but at this point we realize that our definition of essentiality we use so far is actually too strict. Because in literature there are two other residues that are known to be essential, and we do not find these residues here. So we went back to this type of plot, and then if you take some time to look for residue 78, and also some time to look for the residue at position 238, you will see that both of them indeed can mutate to different amino acids, but only to one other amino acid. And if we go and take a look at which amino acid this actually is, it's an amino acid that has very similar properties to the original amino acid that is present. So meaning that some residues that can be replaced by other amino acids can still be essential, which is why we expanded our definition a bit, and we now also define potentially essential residues. And we say that these residues, they can still mutate to other amino acids, but only to a very small number. And for now, we put the threshold at 3. And then we generate this new list of potentially essential residues that now also include the 2 residues that were missing before. Also very interesting to see is that we now include these residues that are shaded in some darker gray that were not described to be essential for protein function, but that we do know are involved in substrate binding, and so therefore are probably important for LPXC. And once again, we also find a lot of new ones, which we are very excited about to explore further. Also, if we plot these potentially essential residues on the protein structure, they are shown now in orange. You see that a lot of them group around the active site. Some of them are located elsewhere in the protein. Does this mean that they are for sure not essential? I think no. I think for these we have to go and manually check if this is the case. And for example, I want to highlight the residue on the far left in orange, which is as far from the active site as you can get, but still I believe this residue is for sure essential because this is actually the methionine at position 1. So in this case, for LPXC, this methionine, the start codon that encodes it, can still be mutated to some other alternative and less efficient start codons, but definitely not to a lot of different ones, explaining why we list it as a potentially essential residue. So to formulate a conclusion to this part of my presentation, I would say that the Onyx platform was quite capable of generating the saturation mutagenesis library, where we get a saturation level of as high as 90%, which I believe is quite impressive and is definitely high enough to start and identify essential residues. And indeed, we have successfully used it to identify residues that are involved in catalysis of the LPXC protein and also in substrate binding. And although this does not apply for LPXC, which functions in isolation, I strongly believe that the same approach can be used to identify protein-protein interactions and allosteric regulation sites if this applies to your favorite protein. Now, obviously, this new information we discovered, we want to use it to extend insights into protein function and hopefully support in the design of LPXC targeting drugs. And now, before I end my talk, I want to talk about this data a little more, but from a completely different perspective. Because while looking at plots like this, which you've seen already a lot now, I realized that in fact there are a lot of positions in the protein that will accept just any amino acid. And I used to think of proteins as these molecular machines that were fine-tuned to perfection by millions of years of evolution, and that even the slightest change would lower the efficiency of this protein. But perhaps this was kind of a naive view, because clearly it is not the case. And this becomes even more clear when we plot this information like this. So what you see here is not just information from the LPXC library, but actually from all libraries we have. And on the x-axis, you see the number of successful amino acid substitutions at any one position. And then you see that in over 40% of all positions, the amino acid that is present can be replaced by anything or at least all but one amino acid, which to me was quite surprising, and it highlights the high flexibility that any one position has in the amino acid tolerance. But of course, this is a very global view. We can zoom in a bit more. Previously, I've urged you to look at this plot vertically, but of course we can turn it around. And if you look horizontally, I think one thing that will pop out, obviously, is that the proline residue is not well accepted because it has a lot of white lines there. And of course, indeed, this is known from literature, and we can capture it also more clearly when we change the plot. So here you see that the proline, the one in orange, indeed performs the worst. In less than half of all cases, proline insertion into a protein is accepted, meaning that in more than half of all cases, if you insert a protein into the, if you insert a proline, into the protein, you will render the protein dysfunctional. And proline performs the worst, and the second worst is arginine. Of course, there's also the other side of the spectrum, with alanine performing the best and cysteine being a close second for us. Now, of course, there's much more information to be gained out of this. For example, we could look at the success rates of amino acid incorporation in function of the secondary structure, or how it behaves differently in the different libraries. And these are, of course, all things we are doing. So if I can give you one take-home message from this presentation, I think it should be that the saturation mutagenesis libraries simply contain a ton of information that is highly interesting and is a lot of fun to play around with. And because it contains so much information, I think it's a very nice starting point for the characterization of any protein, for example, also your favorite protein. And with that, I would like to close my talk and thank the people that were and are involved in this project, and of course, the people who gave the money, which we all need. So with that, I will stop talking, and if you have any questions, we are very happy to answer them.
And thank you so much, Dr. Dewachter and Dr. Klitten, for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. We already have some great questions coming in from our audience members. Our first question, how is Inscripta advising customers as to how to handle the pull of edited cells once it comes off the instrument?
Yeah, so, okay, thanks. This is Laura here. I can take the question. So obviously it's very important when you have this pool of
And I
edits.
think you might have to unmute because we don't hear you.
I am unmuted.
If you were trying to answer.
Hello? Hello, can you hear me? Do you hear me now? Hello?
And
Again? Hello?
I want to thank our audience.
Do you hear me?
Oh, perfect.
Do you hear me okay now? Do you hear me? Okay, so I will answer the question as good as I can. So basically what you have to remember is now you're working with libraries and your edits will be different. So basically the different edits that you're now generating need to be very cautious of working with this library. So we do also offer guidance for how to work with this library to make sure you have the right distribution of your library when you work downstream with your cells. So we are offering guidance for how to handle these cells downstream of the editing.
Our next question, does the Onyx system support editing on plasmids?
Yeah, so I can take that as well. This is Laura here. So great question. So this is a genome engineering platform. So the Onyx is working on the genome level. So right now, this is not it's not something we do. We don't edit on the plasmids, but we do that on the genome. So the advantage of having that is that you're not introducing as such a plasmid that you need to maintain further downstream when you're working with your cells. We're actually introducing the edits right on the genome so that you have already incorporated at the specific place and the specific locus where you want it already from the start.
Thank you so much. These are great questions coming in by our audience members. Now tell me, what exactly does a barcode do? Can you please elaborate on that for our audience?
Yeah, so I can take that as well. Yeah, so great question. So basically the barcode enables you to very quickly and very easily track what's going on with your cell population. So the barcode is located on the plasmid. So as long as you maintain the plasmid inside the cells, then you will be able to use this as a proxy for what's happening on the genome level. And this enables you to very quickly, for example, if you're doing like pooled growth selection, if you want to look at a growth phenotype, then you would actually know very quickly by looking at the barcodes which cells were enriched and which cells were depleted, so which edits were actually beneficial for my specific phenotype and which were detrimental for the phenotype. So the barcode is really giving you a very like easy access to a lot of very valuable information so that you can start like digging into the data very quickly from running these experiments.
Thank you so much. Now, our next question, what percentage of a library pool is unchanged plasmid?
So I'm not completely sure I understand this question. I think I'll have to answer that like by email because I need to dig a little bit what's probably meant, or maybe the person who asked the question can like elaborate a bit on what's meant by this, because we do use a plasmid system to do the edits, but the edits are done on the genome. So I'm not really sure what's meant with like unchanged plasmid. Sorry.
Thank you very much. These are great questions. We have time for a few more. Now, how long an experiment such as the one by Liselot take?
So yeah, let me answer this. So it depends, of course, on what you're talking about. If it's just the editing, I mean, using the Onyx platform, I'm not 100% sure, but I think now this is definitely done in a couple of days. And then after the editing, all we basically did was grow the libraries and then sequence them. And obviously, this can go quite quickly with Illumina sequencing, and then everything just depends on how fast and how well you can analyze your data. So I think really, if everything is optimized and streamlined, in a week you have your data.
Thank you again. Now, your HTP technology generates a lot of samples. It would be difficult to process all the samples. Do you have HTP solution for enzyme expression and assay?
Yeah, so thanks. I can take that question. So Inscripta does not, we don't like provide downstream the testing capacity, but we do acknowledge it's very important when running these big libraries that you need to balance your generate and the test. And we do also have some guidance on how to best do that. But it's basically also a matter of discussing what's the specific project you want to do, what's your aim with this project, what you want to achieve, and then find the best downstream solution to actually do the either screening or selection, or to monitor the different genotypes you had and look at the phenotype. So it's a great question. It's very important to look at the whole experiment and balance those two, like the build and the test part of your experiment. Definitely.
Thank you again. Now, will this platform be available for human cells anytime soon? Interesting question.
Yeah, so yeah, I can take that as well. So Onyx has been developed for microbial systems. So this is a microbial platform that works with E. coli and Saccharomyces cerevisiae. Inscripta is working on a mammalian system that has been announced previously, and unfortunately I can't share more details about that, but you have to follow Inscripta on social media and look at our website for when we can share more details about that. Yeah, but Onyx is for microbial.
Our next audience question is asked, I'm very surprised that the cysteine substitution is tolerated. Is it possible that it is not true for all proteins?
I think it's definitely possible. Already when we look at our 4 different libraries, we see that there are some differences between them. However, cysteine for all of our 4 libraries pops up as one of the best tolerated amino acids. So why is this the case? Obviously, I don't know, but it's just what we see and what is happening and what is biologically tolerated, apparently.
Thank you very much. Now, did you try multiple synopsis mutations at the 265 site to make sure that the site is not simply, let's say, uneditable?
Yes. So before we started, obviously we did not know that this would be a problem to edit this position. Now, in retrospect, of course, we wish we would have included a couple other synonymous mutations. We will not repeat the whole library editing process, but we will go in manually to try and change this site to a synonymous mutation, just this one mutation, and see if it works. And I'm quite confident that we will be able to make a synonymous mutation, yes.
Thank you very much. And I want to thank our audience for these great questions. We have time for about 3 more questions before we close today. This next audience member says, thank you so much for your great presentation. Do you have any explanation for why proteins are more or less tolerant for certain amino acid substitutions? Example, pro, arg versus ala, cis.
Yes. So, for example, if I take the most clear example, I think of the proline residue, it has a very specific structure. It really does not look like any of the other amino acids. So it is also known to break up like alpha helices and beta structures. So it seems quite logical that when we introduce a proline into a protein at a random position, that it has a higher chance of disturbing protein function. From the other hand, for example, alanine has a very, let's say, modest structure, so it's probably tolerated better. Of course, if you have an amino acid that performs, for example, a catalytic function as a proton donor or whatever, probably alanine would also not work very well. But in general, structurally speaking, this is better tolerated, yes.
Thank you very much. Now, are there residues that are highly tolerant of changes also the most variable across population of E. coli or other bacterial species?
I think that's a very interesting question that we are also asking ourselves, but unfortunately we are not yet at the stage in the project where we have examined this. But I think it's a very good remark, and I believe that the most tolerant positions would also be the most variable. But until we go look at it, there's no way to be sure.
Thank you very much. Now, how do we know a residue affects protein function or just affects protein folding process and causes a low expression level when the mutant was eliminated from the selection?
Well, I believe if a mutation changes protein folding or protein expression, it kind of also changes protein function. But I get, of course, the question, you're looking for residues that are more directly involved in the functioning of protein, for example, catalytic residues. And so I think just by looking if a mutant is present, yes or no, there's really no way to say. But I think a more in-depth downstream analysis can answer this question. So looking at whether or not this residue is located at the active site, if it's located at the surface of the protein or more inside the protein. Of course, if it's really buried deep inside the protein, most likely it's about protein folding, etc.
And this is our last question we have time for, and it's a clarification of the previous question. In the final mutation pool, what percentage of cells do not have a change in the target? In other words, what percentage of cells do not have a variant?
Okay, great.
Go ahead, Laura.
I can take that. So basically, I think that's kind of follow up from the plasmid question. So of course, it's important to think about that you're introducing a plasmid into the cell, and then when the plasmid is there, so there's like an editing efficiency or transformation efficiency. And actually, the Onyx is has a very high transformation efficiency, approaching almost 100%. So basically, all cells will have a plasmid. And this is because of the microfluidics that we developed, that this is so high. So that's kind of solved. And then, of course, you also have the actual editing, which will happen on the genome. And obviously, there will be some that are kind of unedited, or some will have what we call like not complete, or because we work with the terminology of Onyx edits, which is complete and intended. So this means that the design and the edit that happened would have to be exactly as designed in the Inscripta designer software. So there will be a percentage of cells that will have this, and then we have a percentage of cells that has like wild type on the genome. And the specification for the Onyx platform is that we see 30% or more, like the fraction of cells that has a complete and intended edit. That is 30% or more. But often we see a high as up to like 75% of the cells. But I would really, if you are interested to know more about the specifics of the technology and how this actually works and how to work with this downstream, please go to our website, and also contact us to get more information on the specifics of these matters.
Thank you so much, Dr. Dewachter, Dr. Klitten. Do you have any final comments for our audience before we go? Dr. Klitten, I'll start with you.
Yeah, okay, thanks. So I just want to say thank you so much for attending this webinar today, and please don't hesitate to reach out to me if you have any questions whatsoever. I'm happy to help also set up follow-up calls, and go to the Inscripta website, and you can see a lot of the great resources we have there for more detailed information. Yeah, so that's all for me. I don't know if you have any final comments, Liselot, you want to share?
Just that I was very excited to be able to test this technology, and I think we really got some very, very nice data out of it, and I'm very enthusiastic about it, yes.
Thank you so much. And Dr. Walker, would you like to add some final comments?
No, I'll leave it at this.
Oh, thank you so much. I apologize. Thank you again. I want to thank you both for your time today and for your important research. And I'd also like to thank LabRoots and our sponsor Inscripta for underwriting today's educational webcast. And before, I want to thank our live audience for joining us today and for their interesting questions. Just as a reminder, questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay safe, and stay healthy. Bye-bye, everyone.